Okay. Yeah, so thank you for accepting our invitation to talk. Please take it away.
Thank you for invitation to this seminar series and giving the opportunity to talk. So I'm Shuntaro Amano. I'm now doing actually a postdoc at University of Strasbourg with Dr. Julie Rangason. And my interest has been centered around the synthesis and analysis of lifelike systems. And today I'm going to talk about my works during the PhD and postdoc with some future prospect about autocatalytic phenomena in ensemble of molecular machines. So as you already know, there are two types of systems in the world: equilibrium systems and out-of-equilibrium systems. Equilibrium systems are static and they don't show macroscopic changes over time, but vast majority of focus of chemistry has been on making new equilibrium systems like drug, plastic, host case complex, many other examples. But recently, synthetic chemists are getting more and more interested in making out-of-equilibrium systems. They show dynamic macroscopic changes over time. And the typical example is, of course, light. And they show interesting functions such as motility, adaptation, evolution, and even information processing. So we're trying to mimic such interesting behavior. And one of the fundamental mechanisms for out-of-equilibrium systems is Brownian ratchet mechanisms. They rectify random Brownian motion at the microscopic scale to generate directional motion, just like macroscopic ratchet mechanisms do. And they are the fundamental mechanism for example, molecular motors, but also for other types of systems, such as self-assembly out of equilibrium, active transport, and so on. And when I started my PhD in 2017, the link between the theoretical study and experimental endeavors was not very strong. So consequently, the experimental efforts tend to lack general design strategy for Brownian ratchet mechanisms. So finding out such general design strategy was one of my interests. But my background is synthetic chemistry, so I started from experimental projects. And what I was interested is autonomous molecular machines. So they are the molecular machines that operate progressively in presence of energy source. So for example, the left one is kinesin, which is a Walker molecule operating in our cells to transport cargoes. And the middle one is a light-driven synthetic rotary motor. And the right one is a chemically driven synthetic rotary motor. But still, the examples of such synthetic autonomous molecular machines was quite limited. So I was wondering, can we make other autonomous molecular machines? And that's what we did. So this is an autonomous molecular pump, which pumps up this blue macrocycle by consuming this chemical fuel. At the beginning, there is only macrocycle and pump, so nothing happens. But once the fuel is added, so this green molecule, this molecule can react with the pump through the cavity of the macrocycle, like this, to form an interlock structure. And then this macrocycle can be pumped to this catchment thread because it's more thermodynamically stable if you look at this energy diagram. And after that, this blocking group can be removed by the base present in the system, and the second cycle can take place. And this cycle can further continue. So you can see the macrocycle moved to this catchment thread, and the blocking group is removed, and the third cycle takes place. So for realizing this system, we first study each step of the operation cycle independently, and then combined everything together. So the first step was the macrocycle take-up. For studying this, we mix these three species and see what happened. And in 10 minutes, the majority of product was this interlocked species, and only 10% of the non-interlocked thread was formed. This is the proof that shows the presence of these interlocked species. So it shows the majority of the reaction take place through the cavity of the macrocycle, which is good for pumping. And the next step is macrocycle displacement. For studying this, we simply dissolve this interlocked species in some solvent and let it equilibrate. When we equilibrated this species with one macrocycle, the distribution between this state, structure, and this structure was 3 to 7. So it favors the macrocycle on this catchment thread. So that was good. And when we equilibrated this species with two macrocycles, then the distribution gets inverted. So it's 7 to 3 now, but still some pumping can happen. When we dissolved this species with three macrocycle, we observed only one species, so no more pumping could take place, and that's because this catchment thread was already full with two macrocycles. The next step in the cycle is the stopper removal, and for studying this, we made these model compounds. One is interlocked structure, and one is non-interlocked structure, and subjected these compounds under the operating conditions. And what we observed was the stopper removal from this interlocked structure was 3 times lower than the stopper removal from this non-interlocked structure. And this is because when this base tries to remove this stopper, it needs to approach this blocking group, but this macrocycle hinders such a process. So that's why it becomes slower. And this is good for pumping again, because it means that after the macrocycle moves to the catchment thread that is supposed to be at this side, it's the removal of the blocking group operated. And the last step is the threading. So for studying this, we use this interlock structure and subjected it under a strong basic condition to remove this blocking group quickly to form this interlock species without blocking group, and then observed the half-life of this species, and it was around 230 minutes, and this is much slower than the time scale of the attachment of the blocking group. So it means after this species is formed, the faster process is the onset of the second cycle rather than the threading of this macrocycle. So it also favors the pumping, so for the next macrocycle. And finally, we combine all of these processes together by mixing the pump, macrocycle, and added a few in a continuous way. And after overnight, we observed not only this species with one macrocycle, but also the species with two macrocycles and three macrocycles by mass spectrometry. So it clearly showed that at least 3 pumping cycles could take place. So we could achieve an autonomous molecular pump. However, the simple design principle for such systems were still not clear. So that was the next topic. And actually, the principle itself already existed, which is known as a kinetic asymmetry. So this is a chemical reaction network for another system. This is an autonomous molecular motor, which rotates around this CN single bond. So you can imagine the bottom half is fixed, and the top half rotates in a clockwise direction by consuming this fuel species and producing this waste species. And kinetic asymmetry for this system, Kr, is expressed like this. And when Kr is larger than 1, this system travels in a clockwise direction. When the Kr is smaller than 1, it's a counterclockwise direction. When Kr is equal to 1, then there's no directional motion. So Kr is a very useful quantity for understanding whether the directional motion can take place, and if so, in which direction. But the problem of this KR is it's relatively complicated expression, and also it includes very small rate constants such as the k minus f, which is the reverse rate constants for this process. And these rate constants are very difficult to measure or control in experiments. So it was difficult to implement this KR for the actual design of the molecular motors. What we found out was there is actually a simpler principle, and we named it Curtin-Hammett asymmetry factor, FCH. So what we found out was that the net directionality of the half reaction cycle, so the part that is shown in red and the part that is shown in blue. So the net directionality obtained from each of these half reaction cycle can be expressed by these quantities. So this one is for the red part, and this second one is for the blue part. And this is a direct consequence of the Curtin-Hammett principle, which is a well-known principle in organic chemistry. And by combining these two quantities, we constructed a new quantity, FCH. And actually, this FCH can tell us the directionality of the system. So this argument itself is quite similar to the case of kinetic asymmetry, KR, but what is interesting for us was that all of these values included in this FCH, so these four rate constants and these two Becquerel constants, are experimentally measurable and quantifiable quantities. So it's much easier to use FCH as a design guideline for the molecular motors. So now we have a better understanding about how to design autonomous molecular motors. We start from a chemical reaction cycle, and then couple it with some mechanical conversion, and then somehow differentiate FCH from 1, and we can obtain directional motion. But actually, this very first step, making a chemical reaction cycle, has been challenging. So chemical reaction cycles, for example, in the case of this molecular pump, is a process that consumes the fuel and generate waste to pump the macrocycle. And here, the conversion of the fuel to waste supplies the free energy to the system to drive the system out of equilibrium. However, it's usually difficult to find compatible reactions that operate under the same environment. And also, even if you can find a good combination, there is often problems such as background fuel decomposition. For example, in the case of this molecular pump, as much as 90% of the fuel gets decomposed without even reacting with the pump. So it's very inefficient system. Various reaction cycles have been developed, but many of them suffer these problems. And what we realized was a simple and general solution for making new chemical reaction cycles, which is to repurpose a known catalytic reaction. So in a common approach, we combine different processes to make a full reaction cycle. But what we did was simply to repurpose a known catalytic cycle. If you compare the general scheme of a chemical reaction cycle and a general scheme for a catalytic reaction cycle, you can see they are exactly the same. So it means we can repurpose a catalytic cycle as a chemical reaction cycle to supply energy. And the advantage of this approach is that we don't need to worry about the compatibility of multiple processes because we already know that each step, the steps in the catalytic cycle operate under the same environment. And also we can suppress the background fuel decomposition because the substrate to product conversion outside of the catalytic cycle is usually much slower than this conversion through the catalytic cycle. So this is our aim. It's not a molecular machine, it's a transient self-assembly but the fundamental mechanism is very similar. So this blue molecule is our fuel, and these two black molecules are the waste species. And this reaction is catalyzed by this red species. So this is the overall illustration of the catalytic cycle. And what we imagined was if different states in the catalytic cycle have different propensity to self-assemble, then we can control the assembly state by simply adding the fuel in a transient way. So if you start from this red state, which has low propensity to self-assemble, and you add the fuel, then this state is converted into this orange state. And this one has a higher tendency to self-assemble. And once the fuel runs out, this orange state returns to the red state, so that this assembly should disappear. So we can form the assembly in a transient way just by adding the fuel. And for achieving this, we did this simple mixing experiment and indeed observed such a phenomenon. And we also confirmed that this red species act as a catalyst for the fuel to waste conversion. This is the result of the NMR. So we could clearly see the signals from this orange species here, which disappeared over time as the fuel ran out. We also studied the effect of pH to this behavior. So at lower pH, the formation of this orange species was not very significant. But if you increase the pH, the conversion to this orange species got higher, and at pH 7, it started to form visible white assembled structures. And at pH 7.9, the conversion to this orange species was as high as 93%. So larger pH, more imminence therefore. This is a microscopic video of this process. So yeah, you can see the formation of spherical aster-like structures. So it's not a random amorphous assembly. They clearly had a structure. And these assembly persisted up until around 1 hour and then started to disappear because the fuel was running out. And yeah, now disappeared. So until this point, I've talked about my previous works. And from here, I want to share some future prospect. And one of the interesting questions for me is, can we synchronize molecular ratchets? So for example, the pump mechanism, the pump system that I shared is not synchronized. So if you look at the system at a given time, it's a mixture of various states. Whereas in the case of an imaginary synchronized system, you start from the pure pump, and after one cycle, all of the pumps have one macrocycle, and then two macrocycles, three macrocycles. So this is a synchronized system. And such synchronization will be important for applying molecular level motion. And possible application can be, for example, in active matter, soft robotics, and so on. And actually, in biology, such synchronization of molecular machines has already been discovered. So I'll share that result. So this is about synchronization of myosins. Myosins are molecular machines that operate in our muscles and contract the muscles. So it consists of this bundle of myosins and an actin filament. And when myosins operate by moving their head, this actin filament is pulled from right to left. And what they found out was that synchronization enables stronger force. And this mechanism is based on simple non-covalent interaction. So this is their experimental setup. They constructed an ensemble of myosins and combined it with an actin filament. They connected this actin filament to this red bead, and they trapped this bead in an optical tweezer. And they also illuminated this bead with this red laser. And when myosins operate, they drag this actin filament and bead into this direction. And when that happens, the optical tweezer tries to restore this bead into the central position. So it exerts a counterforce against the operation of myosins. And the movement of bead is recorded by this reflection of the laser light. And this is the experimental results. When you look closely, they found something interesting. So the free energy of single ATP hydrolysis was around 117 piconewton nanometer, whereas in some cases, the observed work in each step was 120 piconewton nanomole. So they are very similar, and it's more likely that multiple myosins cooperate to produce one step at high loads than the energy of ATP is converted almost in a 100% efficiency into the work, because there is processes like friction and dissipation. And they thought about the mechanism. So this is the six-state model that they had in mind. And the important steps are these two power stroke processes. Power stroke are the processes that cause mechanical motion of the myosin's head and causes the movement of the actin filament. And from the previously obtained data, they calculated the rate constants for these power stroke processes. And important thing here is that these rate constants depend on the strain. So strain here is related to how much the actin filament is moving. So when you look at, for example, K plus 4, when there is no strain, it's very small. But when the strain is negative, so it means when the actin filament is moving from right to left, then these rate constants drastically increases. And it's the same for the K plus 5. And their point was this strain dependence of K plus 5, K plus 4, and K plus 5 actually synchronizes the power strokes. Because when some myosin starts the power stroke, it moves this actin filament from right to left. And this causes a negative strain, and it accelerates the power strokes for other myosins. So there is an autocatalytic or positive feedback loop here. So this is how it works. At low load, the myosins operate in a random way. But once high load is applied, many of the myosins stop at the standby state before the first power stroke, because now there is a positive strain, and under the positive strain, the rate constants for power strokes are very small. But once one myosin accidentally starts the power stroke, then it causes a negative strain, and that leads to the acceleration of the other myosins. And many myosins undergo the power strokes at the same time, so it causes synchronization. So I go back to the first, my first point. So here, synchronization enables the generation of stronger force, and the mechanism for synchronization is rather simple and based on non-covalent interaction. So in conclusion, we realized an autonomous molecular pump, and we also developed some theories for aiding the design. And we realized a simple and general approach for developing chemically driven systems. And through these works, we could strengthen the link between the theoretical studies and experimental works. So the future direction will be to find more practical applications for Brownian ratchet mechanisms, and also to realize more interesting dynamics by, for example, synchronization. So I'd like to thank all of my colleagues during my PhD and the postdocs, and thank you very much for your attention.
Thank you very much.